Hello friends, I am Dr. Suraj Balakani. Today I am delivering the lecture on topic 6 secondary metabolites, biochemistry, and role in plant. So this topic is related with the syllabus of chemistry second year organic chemistry from the subject chemistry of natural products. So in this syllabus and in this chapter, secondary metabolites is one of the most important aspects. In this, we will see how secondary metabolites are classified, how these are synthesized, what is secondary metabolites, and different roles of secondary metabolites in the plant themselves. So we will see the introduction as a part of plant mechanism or uses for plants as well as insects or animals depend on them as well as human beings also. So we will see it one by one. So first we will see secondary metabolites and what is primary metabolites. The first most important thing is that secondary metabolites are in most of the cases derived from the primary metabolites. So in this we will see secondary metabolites derived from the primary metabolites. So first we know that glucose is the primary metabolite. And from this, by the process of glycolysis, phosphoenol pyruvate will form, and from this phosphoenol pyruvate means PVF pyruvate will synthesize. And from this aliphatic amino acids, and from this PCA cycle, aliphatic amino acids, and from these alkaloids are the secondary metabolites. They were formed. And from alkaloids, complex alkaloids as well as more functional alkaloids are formed. In another case, from glucose by the pentose phosphate pathway. Either 4-phosphate will synthesize, and next to that, again, phosphoenol pyruvate cycle is there, and in next second, shikimic acid pathway. So from this shikimate pathway, aromatic amino acids and hydroxyl acids are synthesized. From aromatic amino acids, again metabolites and other secondary metabolites will form. Here, phenyl propanoids also form, and this gives flavonoids and synthesis of complex flavonoids. So this is the basic pathway for synthesis of secondary metabolites. Only for some secondary metabolites, here only simple isoprenoids, complex isoprenoids, alkaloids, and flavonoids are shown in their synthesis by the processes like phosphoenol pyruvate or glycolysis and pentose phosphate pathway, phosphate pathway. So from this pathway, some primary metabolites, secondary metabolites can be synthesized. So primary secondary metabolites from the here. So GA synthesis, GA biosynthesis, GA essential amino acids. So this is pyramid acid, adiabatic acids. This is the resin component, and this is the glycolate alcoholic acids. And here this is proline. So we will see these all one by one. These are the boundary line between primary and secondary metabolites. So main secondary metabolites in the plant. So there are different substances there. We have about 29,000 countries derived from the 3,5 precursors isopentyl diphosphate, IPP. and 12,000 alkaloids derived from the amino acids. 8,000 phenolic pathways are by the malonate and acetic pathways. Main metabolites. So first, we have seen the some introduction about the secondary and primary metabolism. Now we will see in detail about the main secondary metabolites. So most of the secondary metabolites which contains nitrogen. So we will see first nitrogen containing secondary metabolites in these alkaloids. These are near about 12,000. Non-protein amino acids, these are near about 600. Some amines are there. Amines are only 100. And cyanogenic glycosides, these are near about 100. Glycophenolates, these are about 100. Main secondary metabolites without nitrogen. So in earlier slide we have seen about the secondary metabolites with nitrogen, which contains nitrogen atom. And in the secondary metabolite without nitrogen. So in this most major group is the terpenoids. Near about 29,000 terpenoids are there. Some monoterpenoids are there. Monoterpenoids, these are near about 1,000. Terpenoids near about 3,000. Diterpenoids near about 1,000. So diterpenoids, monoterpenoids, terpenoids, triterpenoids, there are different types of terpenes. Triterpenes, steroids, and sulfonics, these are near about 4,000. Phenolics, near about 8,000. And flavonoids, phenolics, they are divided in different classes. Flavonoids are also one of the phenolic compounds. So flavonoids are near about 4,000. Polyoacetyl near about 1,000. Polyoacetyls near about 750. Phenyl protanoids, these are near about 500 units. Compartmentation of secondary metabolism biosynthesis. So mostly in the cytosol, hydrophilic compounds are there, and chloroplast hydrophilic means which are miscible or soluble in the water. And the chloroplast, these are alkaloids, for example, caffeine is used here. And the terpenoids means monoterpenoids are considered. So mitochondria is one of the parts where some amines are synthesized as well as alkaloids also synthesized. This is also one of the part of the plant cell. So in these alkaloids or protobergillins are synthesized. Endoplasmic reticulum, in the hydrogenation steps and nucleophilic compounds are synthesized. Secondary metabolites sequestration. So water-soluble compounds are usually stored in the fuel part of the plant, and lipophilic substances are sequestered into the we can see that they are stored in the raging steps, lactocycles and glandular hairs, trichomes or in the cuticle on the or in the cuticle or on the cuticle. Secondary metabolites sequestrations in the vacuole. So in this slide we have seen about the sequestration in the vacuole. So you can see in this case. So this is not a part of chemistry, it will be the part of biology, but we have to know about the secondary metabolites, how these are synthesized, how these are stored, how these are applied, or how these are used. The water soluble compounds, alkalides, and some primary glycosides, glycosinolates, proteins, anthocyanins, flavonoids, and carbanoids are synthesized in the liquids. And now next part is the second metabolite sequestration in the liquids. So example is given here. Anthocyanin. Anthocyanin is a blue-red flavonoid pigment. They are stabilized. They stabilize in the liquid. Oxidized in the cytosol. And sequestration is a detoxification process. The chemical sequestration into liquids. There is another example is the anthocyanin example. This different when the bronze gene is not ejected, anthocyanin accumulates in the cytosol and can draw inhibitory absence. You can see here two-colored flower displaying on the body. It is a part of nutrition. The location is wire and not with the biomembrane. You can see here fine bundle of trichomes and mainly it is very distributed. It is very simple procedure to get the effects of effects from the plant. The storage of laticifers. Laticifers means we are different types of latex. The latex is a cap mixture. Cap mixture means it is a filled liquid as you can see. Filled fluid. Compound stored in this liquid structure called as the laticifer. Rubber was isolated from it in the past. You can see this. And the composition is typically water, protein, sugar, and ingredients. But often latex has a milky appearance. Long distance transport of technical metabolites in Italian, Solomon, or the apoptotic transportation. So, long distance means technical metabolites can be transferred from the different parts of the plant to different parts through xylem and phloem. So, import glycosinolates are transported from the long distance phloem. Selection of specific glycosinolates will be located into the phloem. Presence of glycosinolates in the phloem provides the difference between insects. Export of glycosinolates from fully expanded leaves and consistent parts. Export of skin tissue and slips and flowers. Host of secondary developed so often give high concentration. Function of the secondary developed often arguments that secondary developed waste product, but this is not totally true. So we can explain by this example. The secondary metabolites are synthesized in the young tissue. Plants are autotrophs and waste products are typically made of heterotrophic animals that cannot degrade their food completely for the degradation. Many secondary metabolites can be metabolized further from phosphate Once secondary metabolite will come in the next step, another secondary metabolite can be found. Both the secondary metabolites are useful. Many secondary metabolites could be metabolized further. Self social and temporal regulation of secondary metabolites via synthesis. So, from this biological activity. Function of secondary metabolites. So, what is the function of secondary metabolites are essential for plant, insects, other animals, as well as human beings. So, difference is the most important role of this secondary metabolites. Another is the extraction. Attraction for insects for the pollination and protection against the herbivores, protection against the enemies. Most animals can now run away and poses and immune system. Plants are attacked by herbivores. As of late, we have seen that most of the uh, plants are full of the herbivores, so this is not a big, big term. So this is generally this is basic. Plants are attacked by herbivores, microbes like bacteria and fungi, and the other plants competing for life, taste, and nutrients. Biotic processes such as regulation functions of secondary metabolites. So first, plant secondary metabolites in the form of mixture, variation in terms stages. The C, one of the most important role of secondary metabolites is the defense against the herbivores, insects, and vertebrates also. Defense against the microbes like bacteria, fungi, or viruses. And that gives the growth, immunization, or toxicity. Another difference is the competing plants, inhibition of germination, and fielding growth. Another work of these secondary metabolites is attraction of the pollinating agents and pollinating insects. It is helping animals, the animals can take the seed from one place to another to attract these types of animals or insects. Root nodal bacteria induce varietal attract and predatory organisms. With this types of functions are explored by secondary metabolites. Examples of plant secondary metabolites and their proposed function. This is one of the visual pollinator attractant. This is routine. This is rotenol. This is the insect feeding deterrent. This is berberine. This is one of the defense toxins. Olfactory pollinating or olfactory pollinator attractant is linolone. Okay. This is defense toxin DINDOA. This is brassaligine. This is one of the antifungal toxins used to the middle synthesize to kill the fungus. Now production of secondary materials for defense against herbivores and pathogens and is not necessary the processes only an infection trigger induce accumulation of secondary materials. Any wound or any damage occurs or any infection occurs on the plant. This is The also synthesis of secondary metabolites, that is known as the induced accumulation. So see here, this is plant. The plant gets wounded or infected from the herbivore or microbe. Due to these wounds or infections, plant activates their defense chemicals, increase the existing defense compound. If there are some secondary metabolites are always synthesized, the defense or the defending herbivores or microbes, their amount or their percentage is increased. Induction of de novo synthesis of defense compounds. Hyperlysins. So that increase the matrix. Function of secondary metabolites. Wounding can lead to release of free segregated compound from the compartment. The mix with the enzyme, often anhydrolase, will result in the production of active form of the chemical. For example, myrosinase and glucosinolates. This is mustard oil bomb, a binary glucosinolase, myrosinase chemical system serious. Glycosinolates break down the production of product isothiocyanates, natural elemental sulfur, thiocyanate, auxiliary begins thione, and iketinoids. So you can see here how this is in this. Is there targets for secondary metabolites in the animal system. So secondary metabolites can attach to the animal different organs of different systems of the animal. First is the nervous system. So here, nervous system of the insects or animal. In this perception, processing, and singular transaction will be damaged, development, muscle or mobility can be affected. Again, digestion, respiration, and reproduction can be affected by the secondary metabolites. Formation in the plants, secondary metabolites. So, the secondary metabolites, these things, this can work in general but not always. So, some herbivores and microorganisms have evolved that have overcome the defense barriers like viruses, bacteria, parasites, and that bypass the human immune system. These organisms develop different strategies of adaptation of secondary metabolites. They can earlier tolerate them or even use them for their diet. Adaptations of specialist herbivores and pathogens. Herbivores, avoidance of toxic plants except host plants, cutting elasticers and region that filled with the secondary metabolites. Non non-resorption or fast intestinal food passage. Reaction followed by detoxification and elimination in urine or other. Hydroxylation contribution is adaptations of specialist herbivores and pathogens. The disruption and accumulation specific compartments cell issues for registration, evolution of intensity, use of plant secretaries in diet, defense against the predators, signal molecules, and morphogens. Signal molecules means preserving pheromone. Adaptations in the specific areas and pathogens, microorganisms, in microorganisms, inactivation of secondary metabolites, evolution of interfaces, co-evolution in metabolites, plant defense, plant taxon, natural enemies, counter resistance, co-evolution in plant secondary metabolites. So, phenomenon is a process due to the its incorporation into the protein that provides the continuity of aberrant polypeptides. With some examples are given here, likewise. Adaptation of specialist herbivores and pathogen to process of co-evolution between plants and their natural enemies is believed to have generated much of the Earth's biological diversity. This includes the chemical diversity. Secondary metabolites in the herbivores. This one is the metaphor given here. Terpene is the most important group of secondary metabolites. In this, the particular number of carbons that can form different types of terpenes. Monoterpenoids have terpenoids, diterpenoids, and triterpenoids. So here, these are examples of monoterpenoids and terpenoids. These are monoterpenoids and terpenoids are here. Expression pattern of the terpene species. Terpenoids are isoterpenoids. The new terpenoids are terpenoids. The main terpenoids derive from the fact that first members of class were isolated from the terpentine. This is the distillate from the pine. That resin. Isopenoid, since isopene is the basic unit of tree-like building thing. That is the isopene of the isopenes. Biogenetic isopene tree. This is isopene and this is lineage. The terpenoids of the plant biological role is volatile and non-volatile. Terpenoids are used as flavors, flavoring agents for traveling, for singing, as antibiotics. They are also used as a hormone. They are used as a membrane lipids, insect attractants, insect antioxidants, mediate electron transport in the respiration of photosynthesis. So, terpenoids and their communication here is given. We have a no problem. Okay. Terpenoids and communication. So, above this is plant. This is part above the ground. This part below the ground. Below the ground. During this part, the terpenes or terpenoids are used as an attachment for the attraction by reflection of fragrance. And again, these are words as a protection, repellent, anti-serum, physical attraction, etc. In the part below the ground, they are also words as a person, attraction as a orientation cues, and below ground protection as the antimicrobial means against the different microbial microbes as well as different compounds. Below ground or the underground terpenes are non-volatile in nature and above ground terpenes are volatile in nature. Again, you can see this part of the slide that earlier we have seen. Here is the isopenoids. So how do you synthesize from glucose, from glycolysis, phosphoenol pyruvate will form from this pyruvate and it gives finally acetylene. Or glucose that can go directly is to do as the glycolose and that gives acetylenides. And from this acetylenides, complex acetylation substance can be synthesized. Use position of carbonides and their precursor. So here, this is gyronyl pyrophosphate, GGPP. Gyronyl pyrophosphate. By the process of gyronyl reductase, this can form the cyclic pyrophosphate. From this cyclic pyrophosphate, by the homogenistic acid, transferase enzyme that gives a 2-methyl 6-cyclic benzophenol. And another from the gyronyl pyrophosphate by the effect of homogenistic acid gyronyl transferase enzyme that gives the 2-methyl 6-gyronyl benzophenol. And finally, this gives the different steps of synthesis, methylation, cyclization, and other methylation, which is the alpha tocophenyl. And from 2-methyl 6-cyclyl benzophenol by the process of methylation, cyclization, and methylation again, there is the alpha tocopherol. Terpenoids, some important molecules are given here in their example. C5 means that are known as the hemiterpenes, for example, isoprene. C10 means niterpenes, for example, limonene. C15 isoterpenes, for example, asbestic acid. C20 diterpenes, for example, gibberellin. C30 nistiterpenes, for example, brassinosteroids. C40 tetraterpenes, for example, carotenoids. And number of carbons above 40 that are known as the polyterpenes. For example, ubiquinone, rubber mixed with the biosynthetic origin, and meroterpenes. For example, cytogenous and vitamin C. Ketone, monoterpenes. Some examples are given here. Linalool, menthol, carbone, metolactone, myrisine, gyranol, 1,8-phenyl, camphor, limonene, ethyl alcohol. So these are some examples of monotoxins. Now, cysteine means 215. Number of carbons are 15. So these are some cyclic and acyclic molecules of the cysteine means 215 molecules. Examples are catechin, germline, karyo, caryophyllene. So these are some examples. How these are synthesized from acyclic to cyclic and cyclic, acyclic to cyclic. Diethyl is 320. So starting with geranium diphosphate by the enzyme is being synthesized. This is casein, and by the process of hexadyne synthase, that is hexadyne. By the process of abidatidyne synthase, that is the labdadienol, propylene diphosphate. And on this the process by the enzyme abidatidyne synthase, that is the abidatidyne. So, trial evidence is the most important and very large class of these terpenoids. Here, 30 carbon units are there. Most important steroids also come under this class triterpenoids. So, we can see here, very simple how this terpenoids are synthesized, how they represent the inclusion reaction, and there are different types of triterpenoids. These are used as the different hormones, steroids, etc. Beta-terpene, most important example of beta-terpene is the carotenoids, means keratin, retinol, or retinoic acid, lycopene, beta-carotene, or xanthocyanins. So, kerosene or beta-carotene, this is the carrot. Biosynthesis is the main compartment. Metabolic pathway leading to the IPP in hypersol, and the NAP pathway leading to the IPP in the plastids. Particularly, malonic, and here NAP and plastids. So here their position and species and their biosynthesis is shown. Biosynthesis of terpenoids, there are three main reactions generating precursors by phenyl transferase. This is one of the enzymes that are found in terpenoids. Terpene synthase reaction, for example, monoterpene synthase and cyclase, and their modification six biosynthesis of precursors. This is dimethyl diphosphate. Dimethyl diphosphate. This plus isopentyl diphosphate. The first organic form is this opening reaction here. One common molecule is formed under taking H plus, so this can form first the geranyl diphosphate. In geranyl diphosphate, again this reaction is the another IDP molecule. In this step, we can say, so one IDP segment is increased, and that gives the here first lamnosyl diphosphate. And again, IPT molecule is added, means there are two vinyl molecules are there. So we can say here vinyl diphosphate. Now we see the biosynthesis of precursor cytosol plastic or methyl diphosphate and vinyl diphosphate are synthesized. Terpene cyclase. We are doing them, then phosphate and multiple products that can be, so these can be shown in this slide. Again, this slide. Terpene cyclase. Terpene cyclase. This is diethyl. So you can see here from this is synthesis of monoterpenes from the one terpenyl carbon or alpha terpenyl carbon. This is from carboxyl diphosphate. This is triterpene, so diterpene from the vinyl diphosphates. So, example are given here. Modifications of monoterpene structures. So, this is first isopentyl diphosphate and the process of enzyme isomerase that gives dimethyl alkyl diphosphate and here vinyl diphosphate. Dimethyl alkyl diphosphate plus isopentyl diphosphate that gives the vinyl diphosphate. By this uh, process of this lignin cycles, if we are being that gives the lignin, lignin, and on the reaction of enzyme lignin 3 dihydrolase with the lignin that gives the trans isophosphoric. And by this process, different types of compounds are synthesized here, step by step. Finally, we get the secondary metabolites using different types of enzymes like menthol, neomenthol, and menthol also. Different processes in the these are again different examples. Discussion here. Production in plant storage, glandrotryptone, labate like mentha, cannabis, so it is equally citrus in the pine tree production and direct emission in the flower phloem tips. The most critical metabolite in the bacil are produced in the cell tip gland. Cell tip gland. One example is shown here. Cell tip gland are typical from the root vessel. This is increased gland. Cell tip. Terpenoids in the cell tip gland. Monoterpene and cysteine. Biometabolic engineering habitations in plants. So in addition to plant alters the profile of the skin as the terpenoids make an important contribution to the environmental studies on their disease regulation. So some examples are shown here. Okay, so these are some examples that are shown for the original different metabolism, individual further modification. This is one here. Thousands of symptoms against surgery. For example, parts reconfiguring the structure. These are some parts like this. Now, so metabolite engineer or metabolite engineering is studied here. It's personal studies for engineered plants. Influence the input behavior. Mhm. Thank you. This is short years. Thank you for joining this video. Thank you. So now we will close the meeting. Thank you.